annual event in memory of her beloved daughter, Susan. So I'm particularly honored right now to introduce our first speaker, Funmi Olopade, who is a professor of medicine and human genetics and is the Walter Palmer Distinguished Service Professor of Medicine at the University of Chicago, where she's been on the faculty for about the last, a little while. last little while. I've had the good fortune to know Funmi for much of that little while through the introduction of mutual friends close to a couple decades ago. And she's been one of the leaders in the study of the genetics and genomics of breast cancer, both breast cancer in general and also breast cancer in terms of the analysis of ancestry. So it's a real honor, Funmi, to have you here at Dana-Farber today and to give one of this year's swirling lectures. Thank you so much.
Yeah, so it's really wonderful to be back here. I seem to come maybe about every 10 years because I have so many wonderful friends here. And really grateful to have been asked to come and talk about ancestry. And I see all my friends in the audience, and I'm originally from Nigeria, so I have to say I'm observing all protocols. But part of the protocol is coming in late at 1:45 a.m. and wondering what's going to happen to you. But here I am. I have no disclosure relevant to this. But what is really top of my mind is to talk to this audience about, sort of how I think about genetics, human race, and, location of where you come from. So for those of you who maybe understand geography, but you haven't really figured out what's happening to genetic ancestry, and we're all talking about black, white, and how do you actually disaggregate data. I want to spend a little bit of time on this map because it sort of came out in 2020 at the sort of height of Black Lives Matter and with student protests and everybody. And so 23andMe published this paper in the American Journal of Human Genetics. And it was really trying to go back to records and then look at how many people left Africa and how many actually disembarked on all the ships. And on my flight in last night, I actually sat next to a sailor who was, going all over the world. And if you study genetics, you understand that no matter where you are, people travel by water, and sailors leave things all over the world. So that's why when we talk about genetic ancestry, if you're on a naval ship, you might have your ancestry all over the world. So when you're thinking about ancestry, I always ask, is it modern ancestry or really what happened as people left Africa? But anyway, what most of us think about right now is black and white, because most of the data in America is based on black and white. And depending on where you are, if you talk about black and white in Europe or in the UK, they actually don't understand it. And so we all get very anxious about the social construct that defined who is black in America and who is not black. I grew up in Nigeria, and we just always said we're black and proud because everybody was black. So pardon me if I spend some time talking about where people come from. This is the big continent called Africa. And it's not one country, it's multiple countries. It has such heterogeneity that I could be black from one country in Africa and be totally genetically distant from somebody from another country in Africa. So that's why I always think about ancestry and then think about geography, because geography is actually more important. So when you look at the Congolese, and there are lots of slaves who came from Congo, and they come directly and they get admixed, and depending on which ship you got on, most of the men didn't survive, or if they survived, they really couldn't reproduce. Talk about the women, they landed, and some of them landed in this place called Bahia in Brazil. And you can see that in Brazil, this little area is almost 95 to 100% African in terms of their ancestry, based on records from the ship. So now if you talk to somebody and you say they are Brazilian, they might come from southern Brazil, northern Brazil. The level of genetic admixture in Brazil will be very different. And based on what countries decide to do with their policies, they may say, we don't want anybody to talk about race because there's not a social construct called race in our country. So there have been lots of things that actually have not allowed us to be able to know how to analyze race ancestry in research. And I use this as an example of, if you go to the southern United States, there are places in the southern United States that are 100% African, and we call them indigenous Africans living in the southern United States. And if you do Ancestry.com or you do Ancestry Informative Markers, many of them can trace their roots back through the Caribbean into Western Africa, okay? And their genetic ancestry may be very different from those who are from Eastern Africa. I'm talking about this because as we talk about violence and ancestry, admixture, migration, a lot of people really want to know why, what's the social determinants of health and what's actually genetic ancestry. So anyhow you look at it, just make sure when you're writing your papers that you know why you want to study ancestry. And I'm using this example of two families, members of the same family. This woman, Jane, developed HER2 positive breast cancer, and she crossed the border to come to me in Chicago, and she got treated for her HER2 positive breast cancer. It was stage 4, she survived, and she became a real advocate for research. Now, how is she related to Diane Artis? Well, she's married to her brother, okay, and they started really thinking about funding research on triple negative breast cancer. Now, you could say they're both artists, but they're married into the family. This is why genetics gets complicated, because as far as they're concerned, we have a lot of breast cancer in our family, but Jane is unrelated to Diane. And then on top of it, this woman who was raising money for us developed triple negative breast cancer, as did her cousin who they were raising money for, and poor Trenes died from triple negative breast cancer, even though she had been the one raising money to fund my research. So that's why I always talk about where you live should not determine whether you live. This woman, stage 4 breast cancer, HER2 targeted therapy, right medicine at the right time. She's fired me as an oncologist because she was cured of her breast cancer. This woman, bilateral breast cancer. By that time, we had learned how to use platinum to treat triple negative breast cancer, and she survived. So what's going on with all this, heterogeneity? And this is where I then begin to think about culture, because here's one of my very first African-American families. We had reported recurrent mutations. I published on 9 families, and I thought we were going to find all the mutations because they're going to be recurrent, like it's been described for Ashkenazi Jewish families. Lo and behold, that would not be the case because there was just a lot of different types of mutations. And then when we could have intervened, and I'm so sorry that Swadlin family had ovarian cancer, and this young woman died of ovarian cancer at 20. So why would she have developed ovarian cancer at 20? And so this woman had bilateral breast cancer, and we could have predicted BRCA1 mutation. And she said, well, I'm not going to have my ovaries out because, if it's not broken, I'm not going to fix it. So how do you see such heterogeneity in penetrance, in age of onset, and why do we think one size fits all? Why did this woman refuse to have her ovaries out? We'll come back and talk about that. But she didn't have her ovaries out, and she subsequently died from ovarian cancer. And so I've been thinking a lot about what can we do to actually mobilize people to act. So, of course, at the same time as we were doing germline genetics, we started really thinking about gene expression profiling. And for those of you who were in the trenches when the North Carolina population-based study started really talking about Lumina A, Lumina B, HER2 positive, basal-like, and unclassified. So Dijon, who is now a professor in the Department of Health Studies, was my first postdoc, who went collecting samples everywhere. And we found that these women were not the same that we described in the textbooks. They were young, and they had overrepresentation of basal-like and unclassified tumors. And we were trying to force them into what is known about ER, PR, and HER2. And if you think about the gradation of what proportion of samples were unclassified among the indigenous African population, and then you can see how different they are as you go from postmenopausal women to white women in Poland and Japanese women, you're going to realize that it's not so easy for us to say black and white. So when we start talking about racial disparity in breast cancer mortality, unfortunately, what we've done is just really reported based on black and white. And so before 1985, there was no disparity between black and white because we treated everybody the same. And as we got everybody to go get more mammography, of course, black women went to get mammography, white women got mammography, and we did make a difference. Then we got tamoxifen, and we got that medical alert that we should give women tamoxifen, and now we're using baby tam to prevent breast cancer. But the thing is, the mortality for black women with hormone receptor negative is twice as high, and it leads to 40% overall mortality gap. And so as we celebrate the successes of amazing drugs and new ways of treating breast cancer, I'm beginning to think, is it black and white? Or is it that there's heterogeneity? So as a geneticist, I always talk about heterogeneity. So this is my 68-year-old white woman in my neighborhood. She's going to come to the same clinic as those black women. And, she has gone to get mammogram every 2 years, as we asked her to do. And she was 2 days late because we blame everybody who go late for their mammogram. And by the time her breast cancer was diagnosed, it was advanced. She died within a year with aggressive triple-negative breast cancer. So the question is, how do we find these women who are going to die from aggressive triple-negative breast cancer in spite of screening them? And so that's really why we've really begun to really think about who is dying. And in my clinic, and we published a series of papers last year, because everyone who comes now, we try to put them on a clinical trial, either they're, neoadjuvant, therapy. And I remember, thinking about neoadjuvant therapy and asking that we should use neoadjuvant therapy for every patient with breast cancer. And I was told, you have to prove it. Well, I proved it because everyone who came through our clinic, we put them on study. So look at black and white women in Chicago. If they have a PCR, there was no difference in survival because we started by making sure everybody had a genomic assay, and then we looked at who was surviving. And then replicated in black and white women in I-SPY, where the way to get in is to have mama print high, low genomic assay. So if I say to everyone, don't treat anybody with breast cancer without a genomic assay, I would say that's a reasonable thing to think about. But everyone says, who's going to pay for it? We can't afford it. But I think based on whether you talk about real world data or any other data, we need to begin to accelerate progress. So our question when we started this Out of Africa theory of early migration was to actually ask the question, what is aggressive breast cancer? And could the burden of lethal breast cancer in Black women be due to differences in the distribution of lifestyle and genomic risk factors? And there have been multiple waves of migration to North Africa, migration out of Africa into India, all over. And there's, if you talk about the Indian Ocean, you will go to South Africa and you hear about colored people in South Africa. And so depending on who is coming, who is immigrant, who is living in your country, you have to ask, what are the genomic risk factors? So that's why we have really focused on genomic risk factors, and we hope that our partners will put in the lifestyle and other risk factors to be able to do better risk prediction. So whether you look at, estrogen receptor status by regions of Africa, if you look at US SEER data, African Americans 64%, US SEER Caucasians, or however we put that, 80%, and then, Kenya, East Africa, 41%. So, everyone is looking at the data based on what is in the published literature. So our first paper was actually the paper we did with Matt's former postdoc, Jordi. And thank you, Matt, for bringing us together. Because what we wanted to do was, let's do exome sequencing and see whether we can use PAM50 subtypes and then compare to TCGA and look at our Nigerian data set with basal. And now we could use RNA-seq and modern technology to divide the data set. And you'll see that in TCGA is predominantly white, but we were able, as a result of some of the extra work that we did, to get our Nigerian data set in there. And then we looked at, interpreting the data. This was published in 2018. And P53 is the topmost gene, then OB2, MYC, all of the usual players. And then we ask, if you look at the mutational signature contributions across race, ethnicity, and subtypes, and you can see that, if you overwhelm your data with predominantly white patients, you may miss a signal. And so that's why in my lab, I always say, well, just let's look at the data a little bit carefully. And you can see that the ApoBec signature, which you tend to see with more estrogen receptor positive breast cancer, is very different from the HRD signature. And the Nigerians were likely to be overrepresented in the HRD signature category. And then I went to Malaysia, and I was talking to my colleague from Malaysia, Korea, looking at MetaBreak, and so Sue Teo said, yeah, we see this TP53 signature and PI3 kinase and GATA3, because one of the things that fascinated us was how many of the Nigerian samples had P53 and then also GATA3 mutation, very different. And so she also was reporting on differences in Malaysia, and we talked about that. So that's the tip of the iceberg, because we only looked at exomes and panel sequencing. And then my husband, who is a pulmonologist, we also had a question around asthma. Why do Black children in Chicago die more likely to die from, asthma? And so, Steve Salzberg called me and said, look, we're sequencing whole genomes from our asthma project, and we see 10% of DNA that's not in the reference genome. And his first question was, do they have an infection? What's going on there? And then, of course, it turns out that with a part of the Asthma Case Control Consortium, they found that, of course, the reference genome was missing 296 billion bases. That's a lot of missing information. So then we, switched to doing whole genome sequencing. And I'm not going to bore you if you're not into cancer genomes, but the basic thing was we were now able to look at 97 whole genomes, tumor normal. Each person was their own reference. And then by that time, we had fewer black in TCGA and fewer white in TCGA. And what we wanted to do was we wanted to reclassify the HRD signature. So this was the single base signature that everybody was using. But by now we had double base substitution and then indels. And I really want to thank everyone at the Broad and everyone working on cancer genomics, because the tools keep getting better. The algorithm keep getting better, and they're going to continue to get better. But the important thing was that we were able to now subclassify our Nigerian samples. We now had more Nigerian samples than in TCGA. And what we found was that it was actually really easy for us using a combination of indel and structural variant signatures along with CORD HRD score. We were able to identify tumors with HRD, and we could also, just by looking at the somatic, infer their BRCA type, because now we had sequenced both tumor and normal. And this is why when we did the experiment, I said, look, this is going to be very expensive, but each person should be their own reference genome, right? Because we have heterogeneity, and we're not going to be able to even understand what we were studying. So I know that we started late. To cut a long story short, we were able to see the signal. We were able to do different clinical subtypes: clinical HER2, HER2 positive, hormone receptor negative, HER2 negative, and depending on your cutoff point, right, you can see different patients in all the different subtypes have HRD signature. So what's driving their mutation? And why do we insist on ERPR Lumina B when there's so much heterogeneity? So it's really beginning to think about what are the driver mutations and what are the mutations that are actually propelling patients to advance very quickly. And we were able to show that GATA3 was mutually exclusive to P53. This morning I was talking to Cornelia about whole genome duplication. By the time you have whole genome duplication, it's really later on, because it's that, copy number changes that you see first. So as we then began to think about how do we think about HRD, how do we think about germline mutation, we've gone on to do germline testing of more than 1,000 cases and 1,000 controls. And you can see BRCA1 is the top gene in Nigeria, BRCA2, and then everything tapers off. You go to Cameroon and Uganda, BRCA1, BRCA2, and then we went to Brazil. And these Brazilian cases, these are the young onset cases in northern Bahia. That's very similar to West Africa. And you can see BRCA1 and 2. Of course, they have a P53 population mutation that everybody wants to study. Very different from African Americans and very different from carriers, where the average age is 62 in their study, and we're talking about women who got breast cancer before 50. So let me end quickly by saying that you're going to hear more about polygenic risk score. You're going to hear more about using polygenic risk score to find populations that are high risk. And you're going to really hear about why you need to make sure your algorithms include people of all different races. This paper has been published looking at clinical genomic features and image statistical features, which vary by site. We happen to have a lot of African Americans in our breast cancer database, and people try to publish whether you can detect race just by looking at digital pathology. And it turns out that it's actually the staining. Our staining was different, and you were just finding the differences based on staining of the slides. Had nothing to do with being black or white. So as we all really think about the future of precision medicine, who we're putting on study, how we want to accelerate prevention, we knew that when you have BRCA1 mutation, your tumor is not going to go through stepwise 2 years, you can go and get screened. And we started using MRI to screen our patient right from 2004 because our patients did not want to have bilateral mastectomy. And then we found that, in fact, the trajectory of BRCA1 mutation is such that you cannot wait to screen every 2 years. You cannot wait to screen every year. You have to find these patients early, and you have to give them alternatives to bilateral mastectomy. So this is my postdoc who developed breast cancer at 30, and she's like, this is unreal. My postdoc who came from Nigeria and said, this is unreal. We've got to figure out a better way. And so they, of course, worked with Dr. Yala, who at that time was a fellow, was a graduate student, and you all have MIRA that you've done as a mammography-based deep learning model. We put our data into it, and now we can use it to get patients into MRI screening. So I'm going to end by saying that we can develop better tools, but we have to make sure we know the patients that we are testing and what we're developing tools for. So let me end by saying, let's reimagine a world without breast cancer, and we're going to go from stratification to precision healthcare for all. And all of you here are going to be part of that, because up till now, it's about whether they're poor or rich. And now I think for precision medicine, it's going to be digital at scale. We're going to be able to integrate both germline and somatic, and hopefully we'll personalize therapies. So thank you all for inviting me. We're demonstrating it that when you do it, you can mobilize an army of workers in Nigeria. And thank you, BCRF, for funding us to do clinical trials in Nigeria. They're ready, and everywhere I go, I really am grateful to be American and to be able to go anywhere in the world, not afraid to be kidnapped. Thank you.
Any questions?
All right, yeah, I got a question from Helen.
Fumi, you have this great whole genome sequencing data. Have you considered looking at HLA differences and how potentially antigens in some of the driver genes could influence the selection of subtypes in different populations?
Oh, yeah. We have a boatload of data. And one of the things that actually I'm hoping for those of you who are looking for postdoc or you want to be graduate student, we are continuing to sequence more whole genomes. Our goal is by the end of this year to have 250 whole genomes and then begin to ask the questions about HLA, so many questions. And you have to understand the mutation signature in the context of the risk factors. And what's unique about our study was we started as an epidemiology study where we're trying to understand reproductive history, and we're trying to understand lifestyle. And every paper we published was null because these women were younger, they had children early, and they developed breast cancer before the age of 50. So what's driving their cancer? And that's really why we think that we have a unique cohort. And every time I speak about them, it's not what is classically in every textbook because the population structure is very different in Nigeria. So yes, if you want to collaborate, we're happy to look at that. That's why I was very excited about some of the work that you're doing.
Chris has a question. I just wondered, you have like amazingly high rates of BRCA mutations in Africa, where Nigeria, and I just wondered what people in Nigeria think about prophylactic surgeries? Are they receptive?
We're going to go on a life-changing journey together
It raises anxiety for different
to explore the most.
just wondered.
Yeah, thank you for asking that question. So I had a very lovely lunch because I said the good thing about the Midwest is we are terra firma. We don't jump upside down about what's going on the coast. So the reason I started doing MRI screening was really because people will come to me looking for an alternative to bilateral mastectomy. They're either from Italy, Catholic, or they are from Eastern Europe. And we were talking with the fellows earlier on that based on even the French women don't want bilateral mastectomy. So I was forced to actually ask the question, will you take an alternative route? And what do I know about the biology and the trajectory? And so when people came to me wanting an alternative to bilateral mastectomy, that's when we started developing MRI physics and trying to look at who needs MRI and who didn't. And up until that time, we heard that BRCA1 does not go through DCIS. Well, it doesn't go through DCIS because by the time we find them, it's already out of the box. So that immune escape that happens between DCIS to invasive cancer, we missed it. And now that we're screening patients every 6 months, that's really what our R01 is about. What do the breasts of these young women look like? What is the heterogeneity of density? And how can we quantify and integrate both the biology and the physics of the breast? And so we are funded to recruit women, and we're going to use PRS as well as BRCA1 and BRCA2. So women who do not wish to have bilateral mastectomy can get on the screening study, and we want to assemble a cohort and see the natural history. Because by the time we found in our first preliminary study, by the time these tumors are 6 millimeters, they already had P53 mutation, which dovetails what we know about BRCA1 null. You have genomic instability, you lose the copy number, and at 6 millimeter, they already had P53 mutation. So what is causing the P53 mutation? Why isn't everybody born with a mutation getting breast cancer? That's really where we're excited to go back and then say, what's the lifestyle? What are the environmental pressures that lead to P53 mutation? And the only way we can study it is if we go back to humans with some, at least, risk prediction tool. And so to answer the question, in Nigeria, when they cannot even get access to basic healthcare, how can we be talking about prophylactic surgery? And that's what a lot of Black women in Chicago told me. They can't get into that because they're just struggling to survive daily. And so we had to get to county hospitals, we had to expand our network, and I'm hoping that we can do this in solidarity, where we're all asking the question, what is the value of our work if we can't get it into the communities that can benefit from it the most?
So, Fumi, wonderful talk, very thought-provoking and inspiring.
Thank you.
I have a question about early or effective diagnosis of breast cancer in Nigeria, more broadly in Africa. So a famous story about modern Africa is that is the sort of technology leapfrog, and the big example has been the cell phone, that people say, in the United States, we had 100 years of landline telephones, and then we go to the cell phone. In Africa, people never built the landlines, and when the cell phone technology came, they went straight to the cell phone. So the question is, could we do the same thing in cancer diagnosis in Africa? And could we go straight to cell-free DNA as a way for cancer diagnosis?
Okay, so I set you up for that because I'm coming back to you, right? The proof of principle is Bloomberg New Economy and Bobby Daly and everybody, and we actually have done the proof of principle that we can find all those copy number changes or the MYC amplification, the HOR2 amplification in cell-free DNA, right? So I don't want them to do any more biopsies. I want them to go straight to liquid biopsy, right? Because if we can do liquid biopsy and we can match it with the treatment, why fool around? So that's why we're going to not worry about anything else, but we're going to do everything that you can get from cell-free DNA. And the idea is, how will we do it, and who's going to fund it? And I think we need partners to be able to do that. So that's why I couldn't meet with Judy this morning, because I was meeting with the Nigerian Health Research Committee, and I was like, you're not going to go back to the future. You're going to leapfrog to the future. And I hope everybody here who has, passion to actually do work in solidarity will not be thinking about black-white, how difficult it is, but will be thinking about how can we leapfrog, because we need to do this work in our own backyard in the United States as well as globally in solidarity.
So yeah, I'd love to talk to you more about that.
Okay.
Been thinking about this. More questions for Fumi? All right, it looks like,
You're going to forever hold your peace, or I'll be here after everybody's completed their talk.
I
And
have one more thing to do.
okay.
Hold on a second. All right, which is we have a plaque for you. And it comes in this box, which is, hopefully they don't stop you in the border of Latvia. But you have this very lovely plaque.
Oh, very nice. Who's going to take a picture of us?
Well, I think. All right, here we go.
But always nice to work with Matt.
Oh, likewise, Fumi. Well, again, beautiful talk. Thank you so much for making this trip to visit us and for sharing your lecture and all your ideas with us. We are very grateful.
All right, I'll take this.
All right, thank you so much. So now it's a real pleasure to, let's see here. Come on up. Now it's a real pleasure to welcome Nilanjan Chatterjee, who is, you were just speaking about Bloomberg, the Bloomberg professor from Johns Hopkins University. Real honor to have you here. Nilanjan's a very famous statistician and epidemiologist, one of the leaders in the development of the field of polygenic risk scores, which is actually right there in his title. And it's a real honor, Nilanjan, to welcome you for this year's Swirling Lectures. Thank you so much.
It's an absolute honor to be invited to give this, be part of this lecture series together with Fumi and Chris, who I know for a long time. And so I changed my title a little bit. I think original title was Polygenic Risk Scores and Precision Cancer Prevention, but I made it a little bit more about the framework. But I will go through the main application is also breast cancer, and where and Fumi went through some beautiful examples. And so I want to start with but I'm going to kind of more emphasize some of the framework that we have been developing, which can be also applied for other cancers as well as other diseases. So I want to start first thanking my wonderful, small but amazing lab I have who have made this work possible, and also a lot of collaborators in Hopkins, outside Hopkins, from Cambridge, National Cancer Institute, other UK institute, 23andMe, we have large collaborations, as well as I want to thank all the participants in the studies from which I got the data and the funding organizations. So, like probably a lot of you are familiar with, like breast cancer, like many other cancers as complex diseases, has a complex genetic architecture, where we knew that from before the era of genomic association studies, through the studies of large families of linkage studies, as well as some candidate gene studies, we had identified these genes like BRCA1 and 2 genes, which contains rare mutations. Is fortunately very rare in the whole population, but individuals who carry this mutation can have pretty high risk of breast and ovarian cancer, and men can have risk for prostate cancer. But then the Genomic Association study era started around 2007, and continue to grow through until now there is about almost 125,000 case and 125,000 control of primarily developed through the Breast Cancer Association Consortium of containing mainly European ancestry individuals. But those studies have now cumulatively have led to about 200 independent genetic variants, the single nucleotide polymorphisms, which are common in the sense that they have at least 1% or so allele frequency in the European populations, but they lead to small effect each individually. But because there are many of them, there is an interest in if we can combine those variants into a score, they might be useful for prediction. So that's what about the polygenic risk scores are about. So what is polygenic risk score? It is basically a construct for an individual which counts the number of risk alleles an individual might carry at different variants weighted by certain effect sizes. And we'll talk about how we choose how many genetic variants to be included in these scores and what are the effect sizes, how do we get their effect sizes. But once those get determined from a genome-wide association study, we can calculate this score for new individuals in new studies to assess individuals' future risk, but also polygenic risk scores and other applications for doing etiologic research. So if we take such a polygenic risk score, let's suppose for the breast cancer, and we evaluate that score for a number of individuals in the populations, or healthy individuals in the populations, we expect sort of this bell-shaped curve because although each individual variants are sort of genotypes, but because we are combined many of them, they will start the distribution is going to look like a normal distribution. And then if we now look at the distribution in a sample of cases, then we expect that in general there'll be a shift rightward, the distribution will be shifted rightward because the cases will have a higher level of score. But in this classic paper by Paul Farrow, he showed that the amount of shift in terms of the mean shift we expect is exactly as the variance of that polygenic risk score in the population, assuming that the score has been well calibrated to the population risk. And which means that if we can add more and more true risk variants in the polygenic risk score, then the variance of the score, this genetic score, is going to increase in the population, and as a result, we'll also get better discrimination of the score between the cases and controls. So what are the factors that are affecting the performance of these polygenic risk scores? So one is very important is the sample size of the training data set, because we are dealing with lots of SNPs with small effects. So to identify those effects, to precisely estimate their effect sizes, we really need sample sizes in the order of tens of thousands of people. And the other guide is how well a polygenic risk score will perform in a particular cancer or a particular disease is this quantity called heritability, which you can think about is the total amount of the cancer risk that could be explained by the type of genetic variants we are studying in genome-wide association studies, say the common set of variants that we are included in GWAS platform. But what is also important is just not the total heritability, but how this heritability is distributed over the number of the different genetic variants. So you can have two different diseases or two different cancers with the same heritability, but in one cancer, if the heritability is explained by a small number of genetic variants, that means that each of those genetic variants have larger effect sizes. Whereas for the other cancer, if it has a larger number of genetic variants involved, that means on average, each variant will have a smaller effect sizes if you want to have the heritability constant. And so this parameter, what is called polygenicity, that means how many sort of genetic variants that might be affecting that risk of the cancer, that also plays a key role in how this polygenic risk will perform. And then, of course, there are all kind of methods have been developed recently for developing these polygenic risk scores. I'm going to give you a cartoon diagram of how these scores are being developed, but some of the operating characteristics of the methods also matters. So if you think about a very simple cartoonist representation of your GWAS data, which is like where we're looking at the summary statistics of SNPs from genome-wide association studies, where you can sort them from the smallest to the largest p-values. And on the right side of what I'm tracking is what is called linkage disequilibrium. That means how the SNPs are nearby each other, they are correlated with each other, and we want to keep track of them because we do not want to double count SNPs, which has highly correlated with each other in our polygenic risk score. And then the first generation of method was very simple, like you focus on the SNPs which have reached the genome-wide significance threshold so that you can be very confident that these SNPs are actually associated with the risk of the cancer, and then also maybe possibly replicated, and then do some kind of pruning exercise to get rid of the correlated SNPs. And that was the first. And then you generate the polygenic risk score by including only those SNPs, but also taking their estimate of the effect size from the same genome-wide association studies. And this was the first generation of polygenic risk scores. The next generation was focusing on this called clumping and thresholding methods, but the idea was that we already know that there is a lot more heritability in genome-wide association studies beyond the top SNPs. So the idea was that why don't we kind of relax that threshold, and maybe we don't have to even fix a threshold. We can vary the threshold, and for each threshold, we can get a particular polygenic risk score, and then if we have a tuning data set, additional, where you can try to see which polygenic risk score gives the best performance. And that led to this whole class of pruning and thresholding type of method. And then there is a whole new series of methods came out where you do not have to kind of hand curate these, finding these independent SNPs or select SNPs. These are under the formula of the Bayesian and machine learning method, which will automatically do that for yourself. Like, they will account for the linkage disequilibrium, they will do the variable selection, they'll do something called parameter regularizations to kind of avoid overfitting the model to the training data set. So that will be automatically happen. And so this is going back to like how does the performance of polygenic risk score depends on genetic architecture of different cancers. So this is a projection we made, a postdoc of mine made in this 2020, where we downloaded summary statistic data from many different cancers and model heritability and polygenicity. And based on that, we are able to predict like how the performance of the polygenic risk score in terms of the discrimination performance is going to increase as a function of sample size. And the sample size are shown in thousands here. And the blue dot is where we currently are. At this point, that is the sample size of the largest genome-wide association studies. And you can see like where we are in terms of getting close to the limits that are suggested by heritability, which is the red dashed line. We are in very different places in different cancers. And that depends on sample size of current study, the heritability, but also the genetic architecture. And for example, breast cancer, because we have been able to correct very large genome-wide association studies, we are kind of, at least if we keep collecting the data on the European ancestry population, we are getting at the point of diminishing utility. We got in most of what we could get from genome-wide association study from the European population by the current study. So we really need to go now into the diverse population to explain more heritability in those populations. So this is where we are in terms of the PRS that was been developed in the European ancestry population, mainly European ancestry population, using the Breast Cancer Association Consortium data, where based on the 125,000 cases and controls, this paper published a polygenic risk score based on this clumping and thresholding method, where they were able to include about 313 SNPs, which have reached a p-value threshold of 10 to the power minus 5, and they're roughly independent across different locus. And that gives that area under the curve in terms of discrimination performance is about 63%. It's a decent one and not a very, very large one. And if you would like the kind of explaining association in terms of odds ratio, this corresponds to a 65% increase in risk of breast cancer per standard deviation unit increase in the value of the genetic score. So we took this genetic score and we understood, okay, this has a, kind of area under the curve 63%, but, 60 area under the curve in my mind, it really does not tell much about what is the clinical utility of this marker as a risk stratification tool. So what we tried to do is that we did a lot of work trying to integrate the risk score together with a well-known classical risk factors for breast cancer to develop integrated absolute risk model for breast cancer. And we did a lot of validation exercise for those models in taking into many different cohorts. So I'll not show all the data, but I will show just quickly that and projection. First of all, these are the kind of the risk factors that went into the model, which is basically the polygenic risk score, but also some of the other risk factors, which are easy to collect. So we haven't included the mammographic density, which is an important risk factor, but that is more harder to collect. But these are kind of classical risk factors that, for example, goes into models like Gale model. And then on top of that, we wanted to see if we added the polygenic risk score, what would be the value of that model. And we developed absolute risk model, and for the in this case non-Hispanic white women in the U.S. population, because the data was available for that population. And then this is a projection of distribution of 5-year absolute risk in this group according to these three models. One is just the standard risk factor model. Another is based on just the polygenic risk score. Another is the classical risk factors. And then we focused on this particular threshold, 3% risk threshold, which is the threshold that is suggested by U.S. Preventive Service Task Force for women to consider to be high enough risk to start considering taking chemopreventive drugs. So we wanted to see, at that threshold, what differences this different model makes. So this is sort of our projections, and again, this is based on the census data and combining various type of health survey data. We estimated that according to the classical model will identify about 3.4 million women in this population to be at that high risk threshold in the US population. But if we use the extended model with the polygenic risk score, we'll identify 2 million additional individuals who will at least classify as high risk according to the model. But that doesn't say the utility of the genetic score, because you're just saying give 2 million additional people this very harsh treatment. So you also have to project how many additional cases we're going to capture in this group. So according to our model, we estimated that we'll capture 100,000 additional cases by giving 2 million additional people this treatment. So what does all that mean? So we boil down this to in terms of, suppose the medication like the chemopreventive drug has about 50% efficacy for preventing breast cancer. So you consider how many individuals we need to treat to prevent one breast cancer. And how does that number changes by inclusion of the polygenic risk score into this criteria. And this was the answer. So it goes from 57 to 48. So you can see there's a decent drop, but you don't see a dramatic drop, which is the reason the reason being that the polygenic risk score doesn't have a very high discriminatory performance. So that's why you don't see a big population level shift in terms of the overall impact. But at the same time, it is adding information into the model. But this also shows we need to keep identifying additional genetic variants, additional risk factors, additional biomarker to reduce that number further. So I'll now move into what is happening in the diverse population setting, both methodologically but also for breast cancer in particular, using some data from actually Fumi's collaborator Dejiang. So we all know that the original Genome Association study, the scene of those was that they are primarily based on European ancestry populations. The representation from non-European population was very bad. And then since the 2008, this paper was published, the situation has gotten a little better, especially for East Asian population. We have a lot more data, but still there is a lot of disparity in terms of sample size in Genome Association study. And as a result, the polygenic risk scores for various complex traits that was developed from the European population, if you look at the performance in the other populations, and this is kind of distribution over different traits, you see a consistent drop across different ancestries compared to the European population performance. And particularly this is worse for the African ancestry populations. There's a dramatic drop in general, which makes some sense because genetically they can be, various reasons, they are most diverse from the European ancestry populations. So this is the same thing people have observed for the European ancestry 313 SNP PRS that was developed based on the BCAC consortium. This is the performance which I reported earlier. They shows an association odds ratio about 1.65 per standard deviation unit. There's some performance drop in the Asian and Latino population, but then when they look at an African ancestry population, I think this is an African American population, there was again a very substantial drop in the polygenic risk score performance. And another problem people have to be aware, and they have noted that when you look at these polygenic risk scores, and if you look at the distribution of these genetic risk scores across diverse ancestry population, you will see this odd shift in distributions. And this shift has nothing to do with the differences in risk in this population. It can be due to various population genetic phenomena like random drift. So we should not interpret these differences in polygenic risk score between population as that is explaining or not explaining cancer risk between the populations. So the idea would be to, we cannot compare polygenic risk score values, at least the raw values, between people of diverse ancestries without doing proper adjustment, of ancestral background. And then again, this has been a very active area of research to how to develop better polygenic risk scores for more target populations like minority populations, using data sets from new minority target population like African ancestry populations, but also borrowing information from the larger European ancestry data. And here, that key technique or the key thing is like you have to borrow information across this diverse ancestry population, because we also know that although there might be some unique alleles that exist in the African populations that are contributing to risk, but the alleles which exist in both populations, they tend to contribute similar risk, so we should be able to borrow information. So there are whole generations of methods has been developed to do that apart, where you based on training data from two populations, you try to kind of borrow information from each other, and there is a tuning step where you try to optimize the tuning parameters and do some ensembling or weighting of the polygenic risk score and for the particular target population you are targeting. And then finally, of course, you have to report the performance of the genetic score in a held-out validation sample so that we really know the unbiased performance of those scores that are coming out through the complex algorithms. So we have contributed to these efforts. Like this is a paper by my former PhD student who spent some actually time also at Harvard and now an investigator, Hao Yu Zhang at the National Cancer Institute. He developed a method called CT-SLAVE that in this paper that we described. But I'm not going to go into the detail of the method. But we also in the paper we also ask some more fundamental questions about, okay, we see the difference in performance, especially in the African ancestry population, the performance is much worse. So how really large genome-wide association study do we really need to catch up with the performance of the polygenic risk score in the other population? Do we really need as large sample sizes as that has been done in the European ancestry populations? And to answer this question, again, we have to go back to what we know fundamentally about the polygenic risk score performance, that it depends on the sample size of the training data, that means we do need sample size from the training data from the target populations. It will depend on the heritability and the effect size distribution and all the polygenicity parameter, but there is not good estimates of heritability or polygenicity in non-European population, in African population. Just today I was having a conversation with Alexander Gusev and Alkes Price. They all agreed that actually, surprisingly, we still don't have good estimates of GWAS heritability in those populations. So we need more of those analyses. Then we'll have a better idea that what kind of sample size we need. And then, of course, it will also depend on the operating characteristics of the method, how efficiently they are able to borrow information from the other population, but also how they are being able to preserve the unique effects from a particular populations. So what we did was that we considered simulations in different scenarios. So here is a simulation scenario. What we did was that suppose we said, like, let's assume that the common variant heritability for, let's say, breast cancer or some trait is the same between the European populations and African ancestry populations. But what happens is that because African has more variants, more common variants, then at a parsnip heritability level, or average effect size is going to be smaller in the African ancestry population. And if this was the true scenario, then we'll see that we're in a very depressing situation. We'll never be, with even a very large sample size, we are not going to be able to catch up with the PRS performance from the other populations. But then we flip it on the other way. So let's assume that on average, at the parsnip level, the effect size is same. It's just the African population has more variants, should have more heritability. So that's the other extreme we considered. And then we saw that this will be actually easier situation where we'll be able to, with a relatively fraction of the sample size of the European ancestry population, and with some borrowing information, we'll be able to catch up with the PRS performance in the other populations. But this is where we left because we did not actually have the data on heritability and genetic architecture in African ancestry population. But since then, more data has become available. So this is the Nature Genetics paper from the African American Breast Cancer Association Consortium, where they reported new genetic variants based on the African ancestry populations. But there is a line that is very important for me, which I was looking for the data, is what is the heritability in the African ancestry population, and the GWAS heritability, the heritability associated with common variant, and they interestingly report the heritability associated with common variant actually in African ancestry population is considerably higher than the same type of heritability that has been reported in European ancestry population. So I thought that is the first piece of good news. But we have to be careful because estimating heritability in admixed population is more complicated when especially you run methods like LD score regression. I mean, we are talking about, again, I was talking to Alkes Price and he was explaining why that might be complicated. So this has to be validated. But I would say this is a piece of good news. But then there was a little bit more good news, more very recent data from, again, Fumi's collaborator and former postdoc, Dejiang. He sent me this data. So they have actually now tried to develop a polygenic risk scores using their data from the African American Breast Cancer Genomics Consortium and combining that information with this, the BCAC consortium data. And what they were able to show that this is the performance of the 313 SNP PRS from the European population, and this is the European performance, and this is the African performance. But now they're developing new polygenic risk scores with their data, and these methods, this particular polygenic risk was developed only using the African data. And they have tried 9, 10 different methods, and this is the best method that came up. And that was the performance of the polygenic risk score. And then this is the multi-ancestry method where they combine the two types of data, and in this case, it happens to be that the best method in this, the winner was our method although there's no guarantee that, that because our simulation showed that different situation, different method can be more powerful. But what is encouraging is that just with a fraction of data, think about it like less than 1/5 of the data compared to European population, this is actually closing the gap of the PRS performance compared to the European population. And this is consistent with our simulation study and the heritability estimate. That we say if the heritability for common variant heritability is going to proportionately increase for African population based on the number of variants, we do expect that we'll be able to catch up more quickly than the other scenario. So the final part of my talk is going to be about, again, the absolute risk-based contextualization of breast cancer risk and as well as mortality, taking into account the complex set of information that goes with the genetic ancestry as well as self-identified race and ancestry, and what we know about the breast cancer rate in the general population, and also mortality that is often not considered. So this is done a lot through using the data from the All of Us study, which if you are not familiar with, it is a massive NIH-led initiative for recruiting people of diverse ancestry individuals from US, and about 1 million individuals. I think already 750,000 people or so have been recruited, and 250,000 of them has been sequenced, and that data is available. There's also another data set where we have genotyping data on a slightly larger number of people. And they have collected all other kind of data based on questionnaire data, different modalities of data, and then in terms of the disease ascertainment, it is a little bit not as good as I would like to be. It is based on EHR-based ascertainment, and at this point, the EHR data was just downloaded at one time at the baseline. So it's all basically is whether somebody has reported breast cancer in the EHR or self-reported to have breast cancer. It's a very prevalent breast cancer status, but also they might have underreporting, because of its EHR, and not all EHR is participating in the study. So it is, in terms of, I would say, ascertainment of disease status, this is not a great study, but it has a diversity. So we try to do as best as we could do with this data. This is your kind of ancestral representation of the, based on the 1,000 genome principal component. We projected that to the All of Us individuals, and then you can usually see the usual clients that you see that is for different self-reported ancestry individuals in US, and the pattern that we see in other studies, you kind of replicate in the All of Us. And then if we look at the distribution of that 313 SNP polygenic risk score, and then remember the problem we mentioned, there could be some weird shift in the distribution. And if we do not adjust for genetic ancestry, you see that it's not as dramatic as the other example. But if you do not correct for this, the kind of the ancestral effect on the polygenic risk score, we might, at the tail of the distribution, we might get a big differences in the who we consider to be high risk. And that's why it's important to account for that. And so this is the polygenic risk score distribution after standardizing by the genetic ancestry. And here you do both mean standardization and a variance standardization to make them more comparable, these distributions. And now after standardization, they look more comparable. And I've highlighted this 5% risk threshold, the highest risk people, because this has been used by the eMERGE consortium, which is kind of developing the PRS reporting as a guideline, as the threshold for breast cancer to be considered alerted that they are at high risk. But this does not account for anything about absolute risk, anything about ancestry, anything about the differential mortality, anything. So you wanted to incorporate all that and see what's going on. So here we are considering a very simple model for developing individualized absolute risk model, just based on genetic ancestry, race, self-reported race, and polygenic risk score. And we are breaking it up into, again, in a very classical way we do in epidemiology and biostatistics, like that there is a baseline risk and there is a genetic risk associated with polygenic risk. The baseline risk can be affected by both self-reported race, identified race and ethnicity, because we do know from studies like SEER there are differences in cancer rate. But we also do not know, that do not have great data. Genetic ancestry might be also associated with baseline risk. And we'll look into some data in All of Us itself, whether that is true or not. And then the polygenic risk score accuracy can be different. We have already talked about that can be, again, different by the self-identified race, by genetic ancestry. There are some data that shows the performance of PRS actually varies more in a continuous manner. So that could be, again, model in terms of looking at interaction between polygenic risk scores and those contexts. So that's the way to account for differential PRS accuracy across populations. So we tried to, although this model should be fitted in incident-based cohort studies, ideally, because that will be a true reflection of risk. But nevertheless, we went ahead and fitted the model, model like this to the all of us data. Remember that the all of us data is the prevalent case control status. So first we look at the, kind of how the baseline risk was different by different self-identified racial groups. And you can see that that's the kind of the pattern we usually see compared to non-Hispanic white, which is the reference group. Most of the other group has a lower risk, which is actually reflected in the CR. The only exception was that for some reason, black cases seem to be overrepresented in the all of us, because these according to the CR, these numbers should be the overall incidence rate for breast cancer is lower in the CR compared to the white population. But otherwise, they match up pretty reasonably well. And then this shows the association of the PRS with the breast cancer in the non-Hispanic white population is exactly what we have seen in the other non-Hispanic white population studies, like for example the BCAC. And then all the negative coefficient, the interactions, show that yes, the PRS is a little bit less accurate in terms of their performance in those other populations. But then in this model, in addition to this, we also included the genetic ancestries, the genetic principal component, and to see whether they has an effect in the actually baseline risk of breast cancer. And surprisingly, we found there are quite a few, we included the PC1, although it was not that strong, but PC4 and PC6, they seem to have a pretty strong association with the baseline risk of breast cancer in all of our study. Now, again, I put a caveat. This is a prevalent case control study, but the case is determined based on EHR status. So I'm not sure that whether this risk is actually true risk or is it something about the reporting of breast cancer is associated with genetic ancestry. And this is beyond the self-reported race and ethnicity reported. But anyway, let's assume that this is actually reflect true risk and this will be replicated in a prospective study. We decided to include this genetic ancestry, these principal components in our model as well. And this shows those three principal components that went into the data. And so you can see that PC4 and PC6, which are very strongly associated with breast cancer risk. So PC4, you see a lot of variation within the Hispanic population. So it's a large variation along the PC4 axis. And the PC6, which is also strongly associated with breast cancer risk, it seems like there's a lot of variation within the non-Hispanic white population in the all of us data. So anyway, this is the final ingredients of our model for absolute risk. So we calibrated the model to the CR incidence rate, category specific to the particular self-reported groups, because we really want to have absolute risk based on incidence rate. We don't care about the prevalent risk, so we wanted to predict incidence risk. And then we also incorporated what is known about the effect of this PRS from previous studies about their differential effect in different racial and ethnic groups. So those all came from not from all of us. All of us is being used for projection. And the only thing that was included from the all of us actual analysis is the effect of the principal components and on the breast cancer risk. We put those parameters in our model. This shows the 5-year our projection of the all of us women in different 3 age categories, like for early age, middle age, and late age, and a 5-year risk of disease incidence, and 4 major ancestry groups. And what you can see across different age groups, and so this one thing I wanted to point out that overall you will see that the high risk group you focus on, these are mostly dominated by non-Hispanic white population, which is expected because they have a higher incidence of the overall breast cancer. And this is true. And another thing that is going on is that notice that within a particular figure, there is a very, this x-axis is the standardized polygenic risk score. So we are showing the risk against that. And you see that there's a steeper curve here for the white population compared to the non-white population. And that is because the polygenic risk score that we went in, that is more strongly predictive of the breast cancer risk. So there's 2 things going on. One is the baseline risk, another is the fact that the PRS is more predictive. But in general, so you can see that the overall, across the different high risk group, you will see that the non-Hispanic white population is dominating. But then we said, okay, let us not stop there, but we know that the consequence of breast cancer is quite different by different ancestry group and different social determinant of health status. We know that the mortality risk, for example, African ancestry women, they can be higher for various reasons. They can have the more aggressive breast cancer, but they can also have the healthcare access issues, and we know that there are differential. So what we now try to project is not only the risk of the disease incidence, but risk of disease incidence and mortality. And what I did a very simple modeling, so that goes into Okay, first you have to have somebody has to develop a disease at a certain time frame, and that is the disease risk model that we talked about. And then we consider the factors that might influence the mortality after the, let's say, breast cancer onset. And here we know from the data that the current polygenic risk score is not much predictive of the survival after breast cancer. And this is true for many diseases and many cancers. But we do know that the survival actually very different by age of onset and also different racial and ethnic group. And those data we got from the CR rate. And there are other factors, can potentially influence this part, like the social determinants of health, but genetic ancestry, but those could be included in the future, but we do not have the data. So you only consider a very simple model where we took just the CR survival rate stratified by age and stratified by the race and ethnic groups. And then what we project is now the 10-year risk of disease incidence and mortality, again, for all of us women in 3 age groups, 45 to 50, 55 to 60, and 65 to 70. And here notice that something is very interesting is happening. Here in the younger age group, you can see the highest risk people are not non-Hispanic white because we have now taken into account the mortality. The highest risk individuals are actually the African American women in all of us. And that pattern starts shifting as you move to the later age, and especially in the 65 to 70, then you can see that the highest risk of mortality is again getting dominated by the non-Hispanic white population. So the point of this whole exercise is what I was trying to show, that there's a lot of work on PRS contextualization in terms of how the PRS accuracy might be differed by different ancestry, different race and ethnic group, but also other factors like social determinants of health. But it is really important to bring in this absolute risk perspective for both the risk of the disease, but also mortality, which is affected by these various factors and also social determinants of health, and see that and to really fully contextualize how the PRS risk affects risk and eventual outcomes after the risk. So with that, I just wanted to conclude by saying that I think one encouraging thing I saw that there is with some sample size from Genome-Wide Association study from diverse population, it seems that we can start closing the gap in PRS performance, at least for breast cancer. We saw that I feel that is encouraging. And in fact, if heritability is any guide, it's possible that we'll have polygenic risk score which performs even better in African ancestry population than European population in the future. But we have to keep in mind is that, if you want to contextualize risk associated with polygenic risk score, we really have to consider absolute risk model. Without that, we cannot consider contextualization. And that will include your self-identified race, genetic ancestry, risk factors of disease onset, but also risk factors for mortality. Once we do that, then we'll be able to see the entire picture, that how starting from a DNA that affects the entire life course of an individual from the disease to the health outcome and eventual mortality. And the great thing is that this is a very standard epidemiologic tool and biostatistics tool like risk prediction modeling, and we do not need to reinvent that part. We just have to use existing tool to that component. So with that, I think I'm happy to conclude, and thanks for a lot of attention, and it's great to be here.
All right, thank you. Miles.
So it's interesting. So one thing that struck me is that there isn't that much added to the absolute risk by the polygenic risk score compared to the classical risk factors for breast cancer. So is that because family history is one of the classical risk factors, and that captures most of what you're finding in the polygenic risk score? So if you leave family history out, family history is there, but family history has a small amount of correlation with the polygenic risk score. So definitely polygenic risk score is capturing additional factor. The main reason that you do not see a huge drop is that the PRS has some predictive power in terms of discrimination statistics, like 63% or 65% increase, but it is not huge. Like in terms of the ability to discriminate cases and controls is not 80%, right? So that is the main reason. It is not, but it is not because the family history is explaining the PRS effect, but it's just the effect is not dramatic. And in this use of the SEER data in the second half, is that only invasive breast cancer, or does it also include DCIS?
So this is invasive breast cancer.
Only invasive breast cancer.
Yeah, Fumi.
Yeah, really great. And so my question for you, when there's a signal, you're going to see that you don't need the numbers. So it's really nice that with all the pool data in Confluence and all of that. So my question for you, and this is really the difference between a clinician and an epidemiologist, and Dijon and I argue about this all the time. So we're talking about a personal risk for cancer, right? And so even though the effect size may not be huge at the population level, when it comes to precision prevention and the personalized risk, it's huge for that woman who is identified, and then you have an intervention. So how do you go from translating in the population to actually thinking about this in terms of contextualizing it in a high-risk clinic? Because the tests are getting cheaper and cheaper, right?
Right. No, it's a great question, and there's a lot of confusion about it. Like it is true that if you look at the very tail of the polygenic risk score, let's suppose the 1% people individuals at the highest risk, 1%, they are actually, those individuals are at a very high risk. Like it could be 4-fold risk compared to the general population, and breast cancer being a common disease, that's a very strong lifetime risk. But then people get confused, then how come it does not make a bigger impact at the population level? Well, the reason is that, like think about BRCA1 as an example, right? It is a rare mutation, but individuals who carry this mutation, they have a pretty high risk, and we have to do something about it. But it will not explain a lot of breast cancer in the general population. So it is the same scenario. So what happens is that those individuals are at pretty high risk, but that doesn't mean that they explain a lot of breast cancer in the whole population. And until we have this higher discriminatory performance of the PRS or other score that we can get by combining PRS, proteins, and other factors, until we get that more larger discrimination, we'll not have this, kind of a Goldilocks situation where we can identify a small percentage of the population which will contain the highest, very large number of cases. So I totally agree, and there's a lot of confusion about it because some people say, oh, look at that, this is a BRCA level risk. And then some people will criticize, what about the AUC? I mean, they're not contradictory to each other, actually. Like, not having high AUC means that there are a lot of cases that are still going to happen outside that high-risk group. But at the same time, you cannot forget that for those high-risk people, they are at pretty high risk. And those have to be done something about it. And if we do identify in the clinic, we cannot ignore that they are at high risk.
So my question may be a little naive, but bear with me. I'm thinking about the genetic instability during somatic evolution of tumors, and it's a little bit like meiosis, right? Because you can enrich beneficial alleles and purge deleterious alleles. So what I'm wondering about is whether anybody has already done or considered doing a joint analysis of the genetic risk with a somatic copy number variation and see whether the somatic copy number variation could, at the risk loci, could tell you something more than you would get just out of the genetic information.
Yeah, yes, people have looking into it. I'm not the best person to answer that, but my understanding is that, so people are seeing very interesting pattern. For example, I saw, not for breast cancer, for example, for some of the hematological cancers, where they are looking into the clonal homatopoiesis, the mosaic changes in chromosome, and based on the, what is called, they call CHIP. And they are seeing that they will find genetic variants, the somatic mutation and the germline mutation, the one that have, if you have the same locus, they have a synergistic effect, So, but I do not know that we have seen that much in the solid cancers, but we have, I think there's some evidence of that happening in more hematological cancer. But again, I'm not an expert on that topic, but that is a lot of question about that. What is the intersecting point about the germline risk and the somatic mutations, the somatic mutation? There's not obvious connection, but most of these common variants, and they're outside genomic regions. We do not sometimes have not been able to even map the genes, because they're so far outside, And so, but there is, in general, for solid cancer, there is not obviously they are in the same locus. They are, generally not that much overlap. But I think for hematological cancer, it's different.
All right, I'm going to take a moment to reply to David's question, and then I'm going to ask you a new question. So just my reply to David's question is about 12 or 13 or 14 years ago, Marcin Imalinsky, when he was in my lab, Marcin's now in the NYU faculty, Marcin looked at this question, and he actually did find enrichment of GWAS-derived risk alleles in somatic amplifications and somatic deletions in cancer. And then he thought that was boring, and he decided to do other things. I don't know, Cece, if you remember him doing this. Yeah, but I actually think we should revisit this question. I think it would be a great question for another person, because I don't think it's boring and you don't think it's boring. And just because Marcin thought it was boring doesn't make it boring. All right, so that's just my reply. And then, Nilanjan, I've got a question for you, because I was actually shocked by one of your early slides, which, and maybe I understood it wrong, but it basically said that 100%, maybe I misread this, 100% of testicular cancer could be accounted for by a polygenic risk score. The AUC was all the way at the top.
Yeah,
And am
90%. Yeah, I mean, the common variant SNP heritability for testicular cancer is about 90% AUC. It will give the area under the curve is about AUC. And in fact, testicular cancer has a very interesting genetic architecture. They have these common variants with pretty large effect, which you do not see in other cancers. Other cancers, sometimes in an isolated, SNPs, you'll see a large effect. But in general, the testicular cancer has a very high heritability, and also not a high heritability, but low polygenicity. That means there are few genetic variants which are explaining a lot of heritability. And as a result of that, even with a small sample size, GWAS, we have now polygenic risk score that can predict testicular cancer with 80% area under the curve.
So back to sort of Fumi's question to you about sort of predictability, doesn't that mean that we should think about implementing clinically in some way this polygenic risk score for testicular cancer? Because testicular cancer occurs mostly in young adult men,
Right.
under 40 or under 45, and although it's not usually fatal, it's still a devastating disease. And
is there interventions people can? What would be the interventions?
so just, I think obviously, surgery, chemotherapy is very effective. Surgery can be very
but for prevention.
Oh, for prevention, you can do testicular self-exam, which is like breast self-exam, but it's easier because it's technically more simple. Right. And making sure that, but I don't think that the uptake of testicular self-exam in the population is actually not that high.
Right.
And so if there was a polygenic risk score and you determined those people at highest risk and made sure that those individuals performed a self-exam or even ultrasound, depending on what the risk is in that population, it might be really powerful. So just something to think about and follow up on.
There are a couple of cancers which are exceptional. Testicular cancer, thyroid cancer, childhood leukemia. They have very high AUC with only a few SNPs.
Wow, even more important with childhood leukemia. All right, I'm gonna turn it back to, and there was one more you said in between. I didn't catch what cancer it was.
Thyroid cancer.
Thyroid cancer, okay. I'm gonna give it back to Fumi.
Yeah, no, I just wanted to say that, cancer is a genetic disease, right? And we just haven't paid attention to the trajectory. And so for anyone in the audience who is really thinking about, how we can do better and accelerate progress, I think these tools are there. They're not perfect, but it's just that we've stayed in the treatment, bucket for a very long time. But if you use the premise that there's a genetic architecture of every cancer, and then we can find them when we integrate germline and somatic genetics. And so the question of mutation signatures, right? Tijona and I looked at, you can look at the signature for HRD and then look for a SNP that predicts HRD, right? Yeah. Because it's basically, we see people in the general population who have a risk as high as BRCA1.
Right.
How do we find that signature? So I think it's really exciting to be able to bring these two fields together now that we're coming together with germline and somatic.
All right, thank you, Fumi. All right, Nilanjan, thank you again. Hold on one second. We're not done. We got to give you...
my plaque, I can't
gotta give you
wait.
a plaque. This is Chris's plaque. We can't give that one to you. All right, hold on. Okay, and just what I said to Fumi. Don't let them stop you from going on the airplane with this.
Thank you.
All right. Yolanda, thank you so much. Really appreciate it. All right. Chris, come on up, and actually we've got, oh, sorry, I walked away with your box. All right, we can't do that. All right. All right, Chris, come on up. So finally, it's a real pleasure to welcome Chris Amos, leader of human genetics at Baylor, one of again, the great people in cancer epidemiology. Has made major contributions to the discovery of most of the major risk alleles in lung cancer, nicotinic acetylcholine receptor, telomerase polymorphisms, probably HLA as well.
It's in there.
Yeah, and lots more, and has really been an inspiration to a lot of us in the field, and has drawn I should say, a big crowd of lung cancer aficionados from near and far to hear him talk. So again, Chris, thank you so much for joining us. We really appreciate you being here and as one of this year's Swirling lecturers. So thank you.
Thank you so much for inviting me. It's a great pleasure to be here and talk a little bit about lung cancer. I thought I would, and it's nice to have my co-speakers. They've done a great job of introducing some of the concepts that I'll talk about, maybe in a little bit more applied way. But and also thanks to David Christiani for attire assistance. So anyway, I'll tell you about that later. But anyway, I thought I would talk about integrating genetics information for precision prevention of lung cancer, and give you 3 stories. So we're in this talk. I'm just describing results from simple case control studies, which are sort of the backbone of the work that we do. So we just compare allele frequencies between cases using markers that have been well characterized between cases and controls, and we just do that millions of times because we use imputed data. So we take the arrays that we have, we compare them to known samples that have been sequenced, and so that gives us a lot of information. It also introduces some challenges that I'll talk briefly about. So, for lung cancer, it's well known that African American males are at elevated risk given the amount of smoking that they undergo, and African Americans are at particularly high risk of adverse events if they get lung cancer. So it's very important to consider diverse populations. And unfortunately, we're in the same situation where most of our data comes from European descent populations. So we do need to do better at diversifying populations that we're studying. We're better at East Asians. Genetic architecture, as Nilanjan was pointing out, and also Funmi, varies by continental origin. African descent populations are more variable. Studying diverse populations provides opportunities for fine mapping that we wouldn't get from just European populations. And so because of that, we really need to expand our sample sizes, and we'll talk a little bit about some of the work. So I'm presenting a study here that we completed in 2022 where we integrated data from different studies across all the populations that we could gather. This is about 70,000 individuals. About 5,000 are African American or African descent. About 10,000 are Asian, and the rest are European. And you can see in lung cancer, the results vary tremendously among the histologies. So that's another kind of challenge for us, is that we have different ancestries and we have different histologies. The thing that's in common across the different histologies are the variants related to smoking. And there's a causal variant in the nicotinic acetylcholine receptor 5 that is the most significant overall, and this is a causal variant that changes signal transduction through the alpha-5 receptor. So that turns out to be quite important in interacting with patients, and I'll talk about that in a minute. for the other histologies, there's actually tremendous variability in the signature. So that's actually informative about the early stages in etiology. And for lung cancer, we have several different causal variants that have been identified. And one of the kinds of predictions that we had made earlier is that for cancers that are environmentally influenced, selection against the variants that cause the disease is less. So, you know, we tend to have more of these causal variants for lung cancer, I think, than we do for something like prostate cancer, which has very little environmental influence. That raises the question we just heard about testicular cancer. Why are there, you know, you would think of all things, testicular cancer would be strongly selected against as a, you know, for genetically, but so why is that? I don't know. Is there some kind of environmental factor that's influencing testicular cancer? It'd be nice to know. So my first story is about an ATM variant that we found in integrating all this work. So one of the nice things about our study is that we have populations from all over the world, including some from Israel and other European parts, East Europe. So we were forced to put this particular variant into our studies by the breast cancer community who were part of this OncoArray project that we did. They are very interested in ATM. We were not particularly interested in ATM, but because ATM is important for breast cancer, it was put on our array. And it turned out that actually this particular variant is very significantly related to lung cancer risk. We also can look across European populations, and when you do that, you see, for example, that Ashkenazi Jewish populations look quite different genetically from many other European descent populations. Bedouins also are quite different. And if you look, for example, in Central Europe, there's not that much variability. And so it's a little bit harder to tell people that are Central European. But from, you know, parts of Europe that are a little more separated, you can easily characterize individuals. And so it turns out this particular variant only exists at any prevalence in Ashkenazi Jewish populations and occurs about 3% of the Ashkenazi Jewish in the world. And in our study, we had a population from Israel that included Bedouins and Israel. It was about 50% of each. And so this allele doesn't exist in Bedouins. It only exists in Ashkenazi Jewish. But it has quite a strong impact on risk. So in our studies, odds ratios for developing cancer, given that you have this variant, range from 2 to 15, depending a little bit on the subsets that you looked at. It's more related to lung adenocarcinoma. And there are homozygous, actually, individuals. So it doesn't cause ataxia telangiectasia. But it has, and if you look at ClinVar, it's sort of unclear whether it's pathogenic or benign, but it is important for lung cancer risk. And also, our collaborators at Sloan Kettering, where there's also a substantial number of Ashkenazi Jewish people, enough to do this kind of study, could show that this variant, when present, leads to loss of the normal allele in the cancer, so suggesting and supporting that it's a tumor suppressor type of factor. So that's one story where ancestry is relevant. And also doing sort of worldwide studies allows you to characterize risk among the different populations. In this case, happen to be Ashkenazi Jewish people, but in different cancers there will be different risk factors. So the other thing to tell you about is that genetic risk is working for lung cancer through smoking behavior typically. And I mentioned this nicotinic receptor 5 variant that is a causal factor related to signal transduction. People with this variant have trouble quitting. because they expect to get a reward from smoking, and if they have the variant, there's a more substantial response to the quitting attempt. And so this slide is showing you that people with the higher risk variants have more trouble quitting and quit later. And there's actually the collaborators at Washington University have been doing clinical trials based on allocating individuals to treatment based on the variant allele that occurs in the individual. And in this slide, I'm showing that if you have the higher risk variant, you respond better to varenicline as a treatment modality. Varenicline is a partial agonist, so people that have the at-risk alleles will respond better to that kind of therapy. So they have done a trial supporting that. Now, what they've liked to do, so this is showing you the way in which they interact with patients. They want to move in, not just based on this variant, but to consider polygenic risk scores. So now we get into the problem of, we've done a GWAS, we have a polygenic risk score, it's only really relevant for whites, and how do we use that information? So this is going with Nilanjan's slide, just showing you that polygenic risk scores are calculated by summing up the risk estimates for each variant, and then also adding in covariates if they exist. In our case, smoking is very important, so we have to model that jointly. And then the question is, why do we care about polygenic risk scores? So there's this famous paper, I think it's from Harvard, showing that polygenic risk scores, for some individuals, generate risk values that are equivalent to what you would get from a monogenic factor. And I think that's true. However, we also sort of have to think about what we're going to do with the polygenic information when we obtain it. So, the problem is, how do you counsel somebody? What do you do with that information? So in one case, I think it's very effective. That's the polygenic risk scores for type 1 diabetes. Those have areas under the curve over 90%. And there's like anti-CD3 therapies that may delay the onset of the disease. So for that particular situation, I think it's very warranted to consider risk score analysis. In our case, we're also interested, the polygenic scores can tell you about biological processes. They may identify high-risk people. And in particular, what we want to do is use the information to change risky behaviors. Smoking is a very risky behavior for lung cancer, so we kind of know what we would like to do. But the way in which we do the work needs to be related to the population in which we're going to apply the result. So we have published a paper a couple of years ago, developing a polygenic risk score. That was with Nilanjan, actually. He helped us to push this through. And we did the work in data that we had from our OncoArray project, and then we validated in the UK Biobank and did projections in the National Lung Screening Trial. And here's showing the range of risk estimates. The model building is probably overfit a little bit. The UK Biobank is underfit because those are healthy people, and they don't have much lung cancer. But still, the highest deciles of risk are elevated enough that you could consider using that for informing people. Again, it's only the highest deciles that are going to be useful, because when you're in the middle, it's hard to tell what the risk actually is because of the uncertainty in the confidence intervals. There is a variation in risk according to age, and also family history. So as you age, the value of the risk information becomes greater. It discriminates better. So that's something we probably should do a little bit better in terms of jointly modeling age and risk. We don't usually do that in our GWASs. We ignore age, and so we're not doing the best job we could. So here we're getting to the point, as I mentioned, the WashU group has a new clinical trial where they would like to provide information to individuals based on their polygenic risk scores about, the value of quitting smoking and adhering to screening. However, we can't really do that with the way the studies have been done, because initially we did the analysis conditional on ancestry. I mentioned that meta-analysis, and then the previous study, and then we combined the data. And so we have a polygenic risk score that was only for whites, so we need to somehow integrate all this information. So the way we are doing that right now is we're using the published studies, doing imputation from the arrays that we're applying to individuals. So we're using a 23andMe array to identify the risk, and then we provide information back to individuals about their ancestry because they find that interesting. It's a reward. But then we have to integrate, allow for the ancestry when we do that. And so we have PRSs for lung cancer and for smoking, and then we have this approach for allowing imputation. And so when we do that, we use, as Nilanjan had mentioned, a mean adjustment and variance adjustment. And you can see this is showing the calibration across the populations. In the top two panels is lung cancer pre and post calibration, and in the bottom two it's for smoking cessation pre and post. And you can see that the calibration leads to much better symmetry among the populations, and it also leads to a better calibrated polygenic risk score with higher odds ratios in the highest group. So this is where we're going. It's not perfect though, so we're now trying to change strategies a little bit. The problem with this, why I say it's not perfect, is right now what we have to do when we're going to work with someone is identify what their ancestry may be, whether it's African descent, European descent, and that's not, people don't want to be classified. They want to be just talked to as individuals. So what we're changing to is a different strategy where when we interact with participants, we collect samples and we evaluate their ancestry jointly with the analysis that's going to be done. The other kind of wrinkle is that we've been using 23andMe for communication with participants. 23andMe is sort of in a state of flux, and we don't know whether it's going to survive. So we're changing to trying to do this with Ancestry.com arrays or being flexible in what we do so that we can preserve in the future. And so when we do that, there are only a selected number of markers that we can retain. So what we're doing now is we're using a principal components based on those common alleles that are present in all the arrays. What we've also shifted away from is doing this analysis conditional on ancestry and just treating the entire population jointly at the same time, and we allow for those principal components that are going to be common across platforms. So when we do that now, this is just showing you that the results are consistent with what we had before, but we also see signals that we know exist in Asians and African Americans, and we also captured some new variants, another nicotinic receptor that we knew was there, we just hadn't detected it before. So the method now works, and when we look at the PRS by ancestry, results are very similar across the groups. There is this little weird shoulder on the left, which is due to some of the arrays not being informative, so we'll see how to deal with that. So the last little story I wanted to tell you is, can we find other factors that influence or increase lung cancer risk? So we know that smoking is the most important factor. Family history is also relevant. But there's this other tool, Mendelian randomization, that allows us to scan for other factors that might be relevant. So we're combining our GWAS studies with lots of other traits that are available from UK Biobank to look for other factors. And so when we do that, we find smoking is the most important factor. That's important. But also vitamin B12. Higher vitamin B12 is not a good thing. And telomere length is also associated with higher risk. So, in the literature or in the popular press, if you take supplements for telomere length, you'll look like this guy. I mean, if only I had taken it a few years ago, I might actually have some hair. But anyway, the idea is that, you can improve your health if you have longer telomeres because you'll, live longer. Actually, that might be true for cardiovascular disease, that longer telomeres are good. But using Mendelian randomization and this kind of results from polygenic types of analyses, you can see that actually longer telomeres are a major risk factor for lung cancer, and more importantly, if you for glioma, they're very important. So you sort of don't mess with Mother Nature unless you know what you're doing. Don't take telomere lengthening compounds. So, there's a concern for cancer. So, in summary, genetic studies of lung cancer identified several causal variants that show substantial variation among populations. This ATM variant that we identified because we were forced to by the breast cancer community, thanks to them for playing such an important role in lung cancer research. Occurs in about, minor allele frequency is 3%, so it's about 6% of the Ashkenazi Jewish population. So it might be considered for screening going forward. Polygenic risk scores can have utility for improving adherence to smoking cessation and guiding treatment options for therapy for those who are smoking. Calibrating multi-ethnic scores has improved the predictive accuracy. For lung cancer, we're still in that, the uphill slope. We really need more individuals to have been analyzed. And we are actually genotyping more people. We're focusing on Brazilians and other populations that have been completely missed in the past. But we're not there yet. Changing the workflow for multigenic polygenic risk scores so that we don't stratify by ancestry is what I would rather that we did. In our case, because we have collected all the samples in the world that are available, we can do the analyses jointly with, allowing for ancestry at the same time as we're doing the analysis, and then we can return the results without arbitrarily classifying people. So I think that's preferable. It's not always practical because, a lot of times you do get the results stratified by ancestry and you'd still like to use that. Still, it's important to consider who you're going to be talking to and how you're going to be presenting the information to them. And then last, the Mendelian randomization identifies additional factors. Having long telomeres is not always good. Don't take telomere lengthening supplements. So, thank you. Oh, and here's our collaborators. Except Nilanjan, he's collaborated with us. Thank you.
All right, Judy.
So thank you for 3 really interesting talks. My question is, do you foresee a time when we'll be able to get our GWAS done? Right now it's very hard to get commercial tests for any GWAS, and
Right.
you feel like they're going to come a disease at a time. But there's no reason you couldn't be genotyped at birth and have some sort of predictive plan for this, although of course they're not big effects. It's easier to explain big effects, and even there we have plenty of trouble. But do you think that's how this will end up, that we'll be able to do this eventually for populations or for individuals
I mean,
broadly?
you can
Will
easily
we have
do-
confluence for everything?
I mean, you
Where are
can
we
easily
going?
do a GWAS. You just send your blood into 23andMe, and they do the analysis. And in my case, I did that, and I found out that I was lactose intolerant, which I knew, but it was sort of nice to know that, you know, genetically. And my wife discovered that she wasn't a celiac. You know, she has problems with gluten, but it wasn't. So, you know, there is actually 23andMe does actually have some information that could be relevant. The problem is that it's not enough, you know. So the real question is, you know, when are we going to do whole genome sequencing? At that point, you know, you get everything that you need.
But you still have to analyze it and then be able to explain it to people. So, I mean, will we be doing this work? I don't think this is a bad thing. You're talking about interventions based on
Right.
GWAS, which is quite different from
Right.
what we've been thinking about.
Yeah, I mean, fortunately, in our case, we have this causal variant influencing, you know, alpha-5 receptor, which affects smoking behavior. So, like in the GWAS information is transferable. It's relevant. I think a lot of times it's not very relevant, you know, for something that you should consider doing. And then the other question is, you know, like if you receive that information, what are you going to do with it? Like we at Baylor, we're doing polygenic risk scores for cardiovascular disease. And, you know, the problem is like the clinicians didn't like it because, you know, then they suddenly have to talk to a lot of people about something where they don't know what the intervention is. So, you know, that's the real question is what are you going to do with the information? So anyway, but I think we're there. I mean, we can get it. It's just, you know, we need decision support for this kind of thing. So.
The first question I had is, do the telomere supplements actually keep your telomeres longer? They don't always, some of the promises don't come true sometimes, especially those guys that look like that. Right. So the question I have is, you know, in taking tobacco smoking histories from people who do and do not have cancer, there's a high degree of variability about how it's done and how accurate it is. You want to talk about how you handle the uncertainty of the information given that it's the dominant risk factor?
The way that our data are collected is, you know, because we use data from many different places. So it's collected as, you know, years smoked, pack years basically. So the problem with pack years is that people start and stop, and so like you won't get that kind of information from these epidemiological studies. It'll be you know, years that they were consistently smoking, and then years since they stopped. But you know, I think that's not bad. The problem with pack years is that the actual effect of smoking is linear in years and quadratic in the amount. So actually it's a little better to be more precise in the way that you model the smoking effects. And that also gets to the problem with vaping is that, you know, it's not the amount that you smoke that's most critical, it's the number of years. And so when people start vaping, they will go back and forth between smoking and vaping. And so anyway, so I'm not exactly answering your question, but...
What about age? Do you smoke a pack a day when you're 20? Is it the same as if you do it when you're 70?
Yeah, so I think if you started smoking when you were 70, that would be okay, honestly.
You already passed.
So, but you know what happens is that you know, it's the more years you smoke, it's an exponential increase in the effect. So, if you start when you're, let's say, 18 and then you quit when you're 28, your risk has gone up, but it's only gone up for the beginning of the exponential curve. So the sooner you quit, the more the increase is going to level off. So, you know, people that, yes, they're at increased risk, but it's nothing like people that have, you know, continued to keep smoking there, you know, for decades. So, yeah.
Yeah, I don't know. A bunch of us are going to start thinking about starting to smoke. I hear it's fun.
Well, I mean, I think the real deal is to actually think about lifestyle interventions, because you can always have a lifestyle intervention, and we can always have 5 milligrams of tamoxifen to really reduce risk. I think the challenge is that we started doing genetic testing and genomic analysis in cancer centers where cancer patients come.
Right.
And I think at some point, if we really are thinking about prevention, you know, the low pass, you know, to answer Judy's question, I always know where the trend is going by the people who come to my clinic. The people who are rich have already sequenced their whole genomes.
Right.
And they are looking for interventions, and they're doing interventions because they actually see where the field is going. So we have a multi-cancer early detection kit that is being tested, right? And I think the question really would be, if we wait for everybody who has a risk to get cancer to show up at our cancer centers, we can't sustain it.
Right.
That's why
right.
we spend 17% of our GDP,
Right.
you know, at the 1 month of life.
Right.
So to answer your question, I would have my whole genome sequenced, and I think that's the idea around all of us, right? All of us was a precision medicine initiative until people in disadvantaged communities revolted and said, you're going to exclude us.
Right.
Right? And by now saying all of us, now he has data and he can model it, right? So I think it's a brilliant idea to think about all of us. So if you want to participate in all of us, sign up, because it's in every community, and the more data we have, the more we'll be able to do better risk prediction. And then the interventions, I think, would happen.
Yeah, I agree with that. And then the other concern or question is, you know, some of our risk profiling has been done in patients that have cancer, for example, and that is going to inflate the risk a little bit, because if there are multiple genetic factors, we know that from BRCA1, where, you know, if you look in families, they're enriched for modifier alleles that increase risk. So getting a cohort together is valuable for looking at the risk, the actual risk. The problem is you need huge sample sizes to get that. So, you know, that anyway, even 100, whatever we're at, it's not enough. So anyway, yeah, go ahead, Nilanjan.
Yeah, no, I just wanted to add a comment to Judy's question about one thing. I think, like, I know we a lot of the time we evaluate the polygenic risk score's utility one disease at a time. But one thing we do forget is, like, it's a tool that is has some economic scalability. That means you get the genome sequence or genotyping data, and then at the same time you can calculate polygenic risk score for all the cancers, right? And it can become a multi-cancer risk tool, and it could be paired with, for example, multi-cancer screening, right? I mean, I know there's a lot of excitement about multi-cancer screening, but I don't hear who are the people, like at what level of risk you have to be that you will be going through multi-cancer screening. And PRS is a great tool for that, because it's a multi-cancer risk stratification tool, right? So I feel like, so other than evaluating one disease, one cancer at a time, we need to show like there is a broader utility, at a multi-disease, multi-cancer prediction, and as a result of the overall impact on the health, not just one disease. And that might convince the insurance company and other people to pay for it, because if we can show more broader utility instead of looking at one disease at a time.
Yeah, I agree with that. I still don't feel like I answered Judy's question quite well enough, but anyway.
One other question, one of the things that we've run into is that the Chinese folks are not anxious to share their data, and they have so much lung cancer amongst the women
Right.
who've never smoked. Do you want to talk about the impact that's having on the international community of trying to sort out some of those issues going on in China,
Well, I
Taiwan?
don't think it's just Chinese. I mean, the U.S. is the best at sharing data, so we present all of our data to everyone, and other countries don't necessarily share as well. So China, my experience with my Chinese collaborators is they actually share very well. They just can't, send the data, the full data. They'll send summary data. So I'm not sure I would just single them out. I would say that the international community is not as mature as we are, and in some ways I wonder if that hurts us a little bit because we make everything available and others don't. But anyway. In terms of non-smokers and others in China, China has its own problem that, smoking is becoming more prevalent there and in the developing world. So there's still, a nightmare coming to, parts of Asia in terms of lung cancer risk. We don't completely know why smoking is less of a risk factor in China than it is in the US. So, that could be partly due to pollution being more prevalent. It could be due to the cigarettes being less harmful. None of that's completely explained. So, but anyway, yeah. And in terms of women and Matt knows a lot about this. EGFR mutations are much more common in Chinese and in Central America. And why is that? There's so much biology.
I don't know this, but I'll make a speculation here that I think this is related to the relative risk of smoking also being lower in China because the risk of EGFR mutations, which are non-smoking associated, is much higher there. And the risk of KRAS mutations, which are smoking associated, is much lower. So my prediction would be this is related to the likelihood of a KRAS mutation occurring and or driving lung cancer, but time will tell.
The other thing, I just want to say one thing, like the signatures, the GWAS identified are quite different in Asians. So we see, VT11A as a risk factor. It hardly exists in Europeans. So, again, there's heterogeneity among the populations. So, anyway.
I have a fairly naive question from the molecular biology perspective in terms of polygenic risk scores. Can you, in silico, reconstruct a lot of the pathways that we know are driver mutations?
Yes.
And then, in terms of therapy sensitivity, because you have this polygenic risk score, but you haven't been able to yet correlate it with therapeutic outcomes or treatments, and can we then see different kind of potential sensitivities?
Yeah, so the answer to the first question is yes, we identify the pathways that we know are related to developed oncogenesis. Those show up prominently in the GWASs. And the answer to the second question is no, that usually therapeutic outcomes are not very well obtained from a GWAS, and I think that's because once someone has cancer, there are many different factors contributing to survival, and so it's been rather underwhelming how well GWASs have done in terms of outcomes, and so.
All right, Chris, really fantastic question, fantastic discussion. I'll take the privilege of asking the last two questions as the host. So the first one is about what you started your talk with, the ATM mutation in the Ashkenazi Jewish population, and you mentioned how it's associated with ATM-LOH. Are there other somatic alterations associated with this as well?
With ATM, I don't, not that I know of with ATM. It's not a cause of ataxia-telangiectasia, and I don't know of other tumor signatures. And actually, from the Sloan-Kettering group, that was the most prominent LOH-related signature among the ones they looked at for germline.
Yeah, very, very interesting. And my second question is about telomere length, because you showed, I mean, it was pretty remarkable. A huge, telomere length is a huge risk factor for many, many cancers.
Right.
Many of which are characterized by telomerase activation.
Right.
Right, and so obviously in lung cancers, you've discovered, GWAS loci for telomerase, as well as this telomere length association. Glioblastoma is another disease with both a GWAS-related allele or alleles and multiple somatic alterations, promoter alterations, as CZ Zhang discovered, translocations, amplifications, and other features. And so it's really pretty striking, the level of increase in GBM. And the other thing that strikes me, with GBM is it's a disease fairly common in otherwise healthy individuals, and perhaps this is related to the telomere length. So I just wonder if you have any further comments on the telomere length and GBM risk.
Well, for lung cancer, David's group just published a paper showing POT1 is involved. So it's not just TERT, it's a lot of the members of that pathway are relevant for lung cancer as well. For glioblastoma, it's very important. I think polygenic risk scores might work pretty well for glioblastoma. Those also have very strong genetic signatures. So anyway, but I'm not sure I responded completely.
Yeah, I was wondering about that, maybe even coupled with the telomere length assessment to start thinking about screening for GBM in those populations. All right, so just want to, before we thank all the speakers, present Chris with his plaque.
Okay, thank you. That's beautiful. Thank you so much.
And we've got the, here we go. There we go. We got the last, I'm going to invite all three speakers to come up with their plaques, and we'll do a last photograph of the three speakers together. Yeah, after all, if your plaques get confiscated in airline security, then you'll have evidence that you really did get this award.
No, I think we'll make it through.
I think so too, but just in case. Right.
Okay.
All right, one without me first. Okay. All right, and one with.
So for those who are staying back, I have a question. Because, I think this idea around ethnicity and, the discussion around who is included and who is excluded, I think we should all be encouraged, actually, that we're having more data. Because you can feel sort of isolated if you think that, in fact, based on genetics justice and inclusion, that we should have had more data on ethnic minorities. And I think this is why I think for today, what I really wanted to convey to whoever would listen is that we have to do this work in solidarity. And we have to go to communities and we have to show the benefit to individuals. And I think what I learned from this symposium really is the mathematical modeling. And everyone will talk about what's in the data. We don't have enough data. We can't do this. Everything is incremental. But what I have observed over the years is that it doesn't have to be perfect for us to make it transformative. And now I think that, I believe that, because the mathematicians are going to get us there. So thank you all.
Thank you.
All right. Well, thank you, Fumi, for those closing words. Thanks to all three speakers for traveling here and for their wonderful seminars. Greatly appreciate it. And one more thanks. Wait. Don't go away. I have to give very special thanks to Claudia Steele O'Sullivan, who is the organizer of this seminar series, and not only the swirling lectures, but our weekly seminars in oncology, and really does a wonderful job with those, including this one. So very grateful to Claudia for these efforts.
Yeah, thank you so much.